Okay, so what this has allowed you to do is completely trash this notion of DNA knows what it's doing. DNA is just the readout, and who knows what's going on? Whatever is controlling the transcription factors, whatever is causing them to do their thing. And this could be a world of influences. And to be absolutely accurate, this requires introducing the word environment. This is environment having something to do with genetic effects. This is going to be a way in which environment is interacting with genetic elements, environment determining which of these are doing what. What would that look like? Sometimes environment could be the environment in the rest of the cell. So you got your DNA there, and your stretches and all of that, and the promoter, and we could do that by now. And something is occurring inside the cell, which activates some transcription factor, and then you go change a genetic event going on in there. The cell is running out of energy. There's various ways to construct sensors. In other words, evolution has come up with a bunch of different ways in which cells can respond to signals of low energy, and that will activate some transcription factor, which will go and bind to a bunch of promoters, which produce proteins involved in taking up more energy from outside the cell, transporting in more, using energy more efficiently. So what we have here is an environmental regulation of genetic effects, environment, the rest of the environment of the cell. The DNA doesn't know what it's doing. The gene doesn't know what it's doing. Events going on elsewhere in the cell is what's regulating what's up. Some of the time, the environment can be even more far-flung. And in this case, it's now events going on outside just this one cell. In this case now, you've got the cell with its DNA, and now instead you have a chemical messenger coming from somewhere else and binds to its receptor like a lock and key, and as a result of this binding, something happens here, which does something here, which does something here, which eventually activates some transcription factor, which goes and does its thing. Now we have gene expression in the cell being regulated by the environment somewhere else in the body. Somewhere else there. What would be a classic example of that? This is what a whole lot of hormones do. Hormones go floating around and they affect cells everywhere throughout the body. By definition, a hormone is a blood-borne chemical messenger. So you could secrete some hormone out of the top of your ear and it will affect things in your little toe. Very far-flung. Hormones will bind to their receptors, lock and key. Most hormones are protein in nature and trigger what's called a second messenger cascade. Jargon. Don't worry about it, just get it conceptually. Activate some transcription factor, deactivate some other and suddenly events going on. 14 counties over there are regulating what proteins are being made in this cell. What would be an example of that? Testosterone, for example. Testosterone secreted from the testes, traveling far and wide and eventually binding to androgen receptors on muscle. And what happens there is, through a pathway like this, turning on the activation of genes, coding for proteins, all sorts of structural proteins that will make that muscle cell bigger. Your muscles are getting bigger thanks to testosterone. Look at this. Events occurring a gazillion cells away in the testes, a messenger here determining what gene expression, what gene activation is happening.